Herzlich willkommen zurück von der kurzen Mittagspause, aber trotzdem herzlich willkommen zurück. Wir starten, um in der Zeit zu bleiben. So welcome back everybody after a short lunch break where we'll continue because in the interest of time. And I'm very happy that we have the head of the ARCA separation science with us this afternoon. Dr. Martin Vogel is here. He was also the former head of the whole analytical division. So thank you very much, Martin, for taking the time. And he's going to introduce this year's Analytical Chemistry division winner. He or she, we'll see. Maybe you know this already from the lineup there. Okay, so Martin, the floor is yours.
Yes. Thank you very much, Carsten, for the introduction, and I'm happy to be here today. So normally Uwe Karst would have been here as a board member of the Division of Analytical Chemistry. Unfortunately, he couldn't make it due to the pandemic, and so I have the honor to replace him here, and that's very nice because I know the awardee of this year. The award we are giving now is the award of the Division of Analytical Chemistry within the German Chemical Society, so we call it then in German Fachgruppenpreis. And this award is for young scientists in the early stage of their career when they are becoming independent researchers and then that they have shown already large amounts of independence. The award for 2022 goes now to David Clases. You will see him within the next minutes. And I would like to introduce the awardee. David Clases, he started chemistry at the University of Münster. He started there in 2009, and so I had him in the first winter term as a young, not as a little student, but as a young student, so I know him very long now. After his bachelor in chemistry, he did also a master in chemistry, also at the University of Münster, and he did the master's thesis in 2014. Afterwards, he came to the Karst group as a PhD student, and in 2017, you did your PhD thesis, you finished it, and you got your PhD for the thesis Trace Metals in Medicine and in the Environment. And during your PhD thesis, you have been to Australia for a couple of months, and you really liked to be in Australia, obviously. So you started a postdoctoral fellow after your PhD thesis in 2017, and you went to the University of Technology in Sydney, where you worked with Philip Dobel for 3 years, and the Australians were very happy with your work. And so you became, in October 2020, you became an independent researcher at the faculty there. And many people were aware that you were very successful there. So you got a DFG Rückkehrer scholarship, so a kind of DFG returner scholarship, and you returned to Germany. The Germans were a little bit slow, so you have been here just for 1 month in 2021. And the Austrians, not the Australians, the Austrians were quite rapid, so they appointed you as assistant professor at the University of Graz in December 2021. And since December 2021, you have been there as an assistant professor. And it's nice to see that you got this tenure track position, and I wish good luck for the next years there. David Clases is author of more than 22 papers in peer-reviewed journals. David got a lot of scholarships during his PhD thesis and also afterwards. He was also very busy in getting some funding and very successful with that. And his research is mainly focused on single particle and single cell analysis by means of ICP-MS, but another field is also speciation analysis. And just finishing the introduction of the awardee with some personal things. So David is also a very creative researcher in the lab. So we had a lot of situations where we were looking for some instruments that should be in a certain lab, and David used it for very creatively in another lab, and we found it then often by coincidence, for example, some balances that had been vanished, and taken by, by David for his instruments. So it was a very good time in Münster, David. So I hope you keep your creativity. And I will now come to the certificate of this award. And therefore, I would like to that you come on stage. And I will read the certificate text. And now I have to switch in German. So the text has been formulated by our division chair, Caroline Huhn, who can also not be here, unfortunately. And I now switch to German. David, die Fachgruppe Analytische Chemie der Gesellschaft Deutscher Chemiker verleiht anlässlich der Analytica-Konferenz 2022 den Fachgruppenpreis Analytische Chemie 2022 an Assistenzprofessor Dr. David Clases, Karl-Franzens-Universität Graz. Und jetzt die Begründung. Ich habe gerade schon viel gesagt, aber es wird auch amtlich jetzt. Der Fachgruppenpreis wird Herrn Assistenzprofessor Dr. David Clases verliehen in Würdigung seiner hervorragenden, originären und zukunftsweisenden Forschungsarbeiten auf dem Gebiet der Einzelzell- und Einzelnanopartikelanalytik unter Verwendung der induktiv gekoppelten Plasmamassenspektrometrie ICP-MS. Hierbei konnte Herr Professor Clases durch die Optimierung von Geräten und Methoden, aber insbesondere auch durch die Entwicklung und Verbesserung von Software-Tools zur Auswertung der großen Datenmengen wesentliche Beiträge leisten, die den aktuellen Forschungsstand der Single-Particle- und der Single-Cell-Analytik deutlich erweitert haben. Auch auf den Gebieten der Elementspeziationsanalytik und des Element-Bioimagings entwickelte Professor Clases leistungsstarke neue analytische Methoden und wandte diese auf aktuelle biomedizinische und umweltwissenschaftliche Fragestellungen an. Seine Arbeiten sind in zahlreichen Publikationen in den führenden Zeitschriften unseres Fachgebietes veröffentlicht worden. München, den 23. Juni 2022, Professor Caroline Huhn, Vorsitzende der Fachgruppe Analytische Chemie, und auch in Ihrem Namen und im Namen der ganzen Fachgruppe herzlichen Glückwunsch, David.
Vielen Dank.
And now I switch to English again, and I hand over the word to David, and David will tell what he has doing over the last years and give a little summary. And David, the floor is yours.
Thank you.
Dauert ein bisschen, aber ja.
There you go. Yeah. Okay, yeah, thank you very much for this kind introduction and for digging up the old stories. Yeah, thanks for not mentioning what I smashed in those times, but yeah. Yeah, before I say anything else, I would like to also thank the GDCH and especially the Fachgruppe Analytische Chemie and the DS as well, Kerstin, Carsten, for inviting me and for giving me the opportunity to present you my data today. So, as Marty mentioned, I just came back from Australia basically, and I'm currently an assistant professor, and over the last couple of years I was given the opportunity to build my own research agenda as well as my own research group. And yeah, I'm quite excited to be here today, and it's definitely a good feeling to have this face-to-face situations again. And today I'm going to talk about analytical tools for the characterization of nano and microstructures. There you go. Yeah, so we have been interested in characterizing small structures, and we started looking at particles which are only a few nanometers large, and then also started studying how they actually behave together, how they organize themselves in nanocomposites, and how they build larger structures. From there, we could also advance onto the microscale and actually show that we could also analyze microplastics by targeting the carbon content, for example. From microplastics, it's actually just a small leap, dimension-wise, for single cell analysis, and we can actually analyze the cells as well. And we can also study how these cells organize themselves in entire organisms, in different anatomical structures, like for example here in the brain, but also in aberrant environments in tissues, for example, in this case, for example, in tumors and the microenvironment.
The common currency for all these topics is basically inductively coupled plasma mass spectrometry and its associated techniques, including single particle analysis and, for example, laser ablation, as well as chromatography. Today, for time reasons, I just picked 3 areas that I would like to discuss a little bit further. So first of all, in the first part, I will show you that we can use single particle ICP-MS to describe quite small and complex nanomaterials. Afterwards, I would like to show you that these nanomaterials also interact, for example, in the environment and build entire new structures. And in the last part, I would actually stray a little bit further away topic-wise and discuss how we have microstructures in biological tissues and how they matter in different pathologies. So first things first, as I mentioned, the core technique for me is inductively coupled plasma mass spectrometry, and today we have already heard about the basic underlying principles here, so I'm going to be rather quick in this case. So in ICP-MS we have a very hot argon plasma which reaches temperatures of up to 10,000 degrees, and whatever is introduced into this plasma gets immediately vaporized, atomized and ionized. So we are generating elemental cations, and these ions are extracted in an ion beam which is then mass filtered and detected. As long as we have here this stable supply of aerosol, we also have a consistent ion yield, and we're getting this steady state signal or plateau signal, as you can see here. This instrumentation can also be operated in the so-called single particle mode, and the principles are quite similar. The main difference is that we are introducing particles individually, so bit by bit. So particles enter the plasma individually, they get atomized and ionized individually and form secluded ion clouds. And these ion clouds can be extracted in discrete packages. These packages then also get mass filtered and detected as well. Unlike before, we're not having this plateau signal any longer. We actually now have discrete pulses, and each pulse correlates to the detection of one nanoparticle. The frequency here is correlating with the particle number concentration, and the intensity of each pulse is proportional to the isotope's mass in the detected particle. Nowadays, we have quite fast operating mass analyzers, and here we're actually fast enough to actually resolve these kind of packages here with several data points. So we can resolve peaks, which we can integrate and then further process afterwards. And then starting from there, we can do some very basic statistics. And the easiest thing you can do is just sorting the intensities that you just integrated by their value. And that gives us a histogram. And here you can see the intensity of the signals that we integrated, and here we're counting how often we detect them. And you can see easily that we have 2 different signal populations. We have one here on the left that corresponds to the detection of background and noise, and we have one here on the right that correlates to our nanomaterials, and of course that's what's interesting for us. So from here we can apply some straightforward calibration approaches and translate that histogram into a model for a size distribution. But it doesn't end there. We can also determine mass distributions. We can have a look at the stoichiometry, stabilities, as well as the agglomeration behavior, for example. There are a few factors which are limiting single particle ICP-MS, and I would like to talk about one especially, which is ion transmission. So that becomes clear when we consider a nanoparticle which has roughly a diameter of 7 nanometers. That particle would consist of approximately 2,700 atoms. In ICP-MS, we typically have an ion transmission which is below 0.04%, which basically means that from these 2,700 atoms, we can just transmit one individual atom or ion towards the detector. And that's actually the reason why ion transmission here is limiting, because if we tried to analyze a particle that is smaller, we simply don't have enough ion transmission to transport at least one atom or ion towards the detector, and therefore they become invisible. So in this case, the 7 nanometers is the size detection limit. So gold particles are kind of ideal particles. They are quite stable. They consist of only one element with one isotope. In reality, we're often having particles which are becoming more complex. And there's an example here. For example, consider a nanocomposite which is consisting of 4 elements with theoretically 5 equally abundant isotopes. In that case, we would be forced to select one of 20 isotopes for further analysis. And in this case, in order to still transmit enough of that single isotope towards the detector, that particle has to be massive, relatively massive. So in this case, it has to be 20 nanometers, and it has to consist of at least 54,000 atoms. So you see, the more complex the particle, the more difficult it is to apply single particle ICP-MS. And that's That's a pity because we have lots of interesting nanomaterials which are currently emerging for various different kinds of biomedical applications for industry as well. And one of these particles are the upconversion nanoparticles, which are rather complex. So these particles have lots of potential for example, drug delivery platforms, for new imaging modalities, but also for solar energy harvesting as catalyst. These particles consist of a yttrium fluoride host structure, and we have lots of lanthanide which are doped inside. However, usually they are applied in very small sizes which are outside the detection limit of ICP-MS. That's something that we worked on to improve, and there are different ways to actually improve ion transmission. And today I would like to present you one way to do it, and that's by manipulating the bandpass of the quadrupole. So here's a short reminder. The quadrupole consists of 4 metallic rods. And on these rods, we are applying a combination of voltages. We have the direct current voltage and the radio frequency voltage, short DC and IF voltage. These 2 voltages cause a modulation of the electric field inside. So for example, if we have an ion, let's say for example 176 ytterbium, that ion starts feeling this modulated electric field and starts being pushed around in it, and it typically follows this typical helix-like trajectory. In order to make it from one side of the quadrupole to the other side of the quadrupole, we can actually only apply a certain set of DC and IF voltages. And to find out which kind of sets are applicable, we can have a look at the stability diagram. In this diagram, we are plotting the DC voltage against the IF voltage, and we get this almost triangular-like shape. And this is called the stability region. Any kind of DC and IF combination inside here gives our ion a stable trajectory. However, we have to consider that we usually don't only analyze or have one specific isotope, we have a bunch of them. For example, in case of ytterbium, we have 5 additional other isotopes, and you can see here that they actually have quite similar stability diagrams. So if I tried to analyze or use a combination of DC and F voltage, which is here, I would transmit 176, but I would also transmit all the other isotopes because here we are in an overlap region. Here, all the different isotopes have a stable trajectory, and I would not be able to tell them apart. So what the quadrupole usually does, it is looking for areas without overlap, and that's in the tips. So in the tips, we are only analyzing one specific mass to charge ratio, and the quadrupole only applies DC and F voltages, which are on this scan line, and it jumps basically from tip to analyze the different isotopes sequentially. So that's where we stepped in. So we actually started to manipulate the voltages applied to the quadrupole, and that allowed us to modify this scan line. And this we called the bandpass mode. So you can see in this bandpass mode, we actually go away from the tips, and we lose the ability to distinguish between mass to charge ratios. But we also win something, and that's ion transmission. And that becomes clear on the next slide. For example, let's have a look at the analysis of gold. Gold is a monoisotopic element. It only has 197 gold. If we analyze it with a standard quadrupole method, we get one tiny signal off here. As soon as we switch into the bandpass mode, we get wider peaks because we lose mass resolution, but the peaks become also much higher, and here we win 800%. The second effect kicks in as soon as we start analyzing polyisotopic elements, for example, ytterbium. You have seen ytterbium has 6 different isotopes. We can use a standard quadrupole method, and we can isolate each isotope if we want to. As soon as we switch into the bandpass mode, we have 2 effects kicking in. The first effect is related to what we've seen for gold, so we have more transmission for each individual isotope. And the second effect is actually the convolution of isotopic signals. And you can see that here in the simulated peak. We also measured one of these signals, and you can see that we get for all isotopes one massive peak, in which we cannot tell apart the different isotopes, but where we have massive ion transmission. And that's also shown here. So we compared the 2 modes. In red, you can see here the standard mode. As soon as we switch into the bandpass mode, we get this peak, and we actually win 3000% of ion transmission. So how is this useful? Well, this is useful in cases where we already have some information on our samples. For example, if we analyze upconversion nanoparticles, we are already quite certain what's in there. We can actually rule out that there are some elements potentially interfering. So if we don't need this, the selectivity, we can actually sacrifice it and win ion transmission or sensitivity. And you'll see that we can do that for upconversion nanoparticles. We can also implement it in elemental bioimaging and also for some other approaches. So in case of upconversion nanoparticles, I mentioned that we have these small particles which incorporate, for example, lanthanides. And here you can also see again our ytterbium. So again, with a standard quadrupole, you can see we can separate the isotopes, but we're not very sensitive. Switching to the bandpass mode, we get extra sensitivity. We win basically 3000%, and here you can see the result. In this case, we were targeting ytterbium in a 20 nanometer sized upconversion nanoparticle. And using a standard method, you can see here that we are not able to isolate this typical nanoparticle signal distribution on the right. It actually merges with the ionic background, which makes it almost impossible to do an accurate calibration. Switching to the bandpass mode, we now have for the first time enough transmission to actually isolate this particle signal distribution here on the right. And we can now do calibrations to, for example, investigate sizes, masses, and much more. And that's what we did in a proof of principle paper. In this paper, we had a look at different kinds of upconversion nanoparticles, different sizes, shapes, compositions, and we were able to characterize them quite accurately between 10 and 100 nanometers. We were able to look at the stoichiometry and even could characterize how they interact with each other just by using single particle ICP-MS. So you can see single particle ICP-MS is quite useful to characterize engineered nanomaterials. And we can do that, for example, for the later application for biomedical workflows, but we can also analyze them in the environment, and that actually becomes a topic with increasing relevance. Next to engineered nanomaterials, there's also a huge amount of particles which are produced incidentally, and they're also released into the environment, and there's a lack of knowledge what they actually do in the environment and how they interact with the biota. So in the second part, I would like to show you that we can also trace nanomaterials in the environment. In this case, I would like to present you a case study that we did in Melbourne. We did sampling 2019, and in this case study, we were interested in different questions, and we did a quite broad sampling. So we were looking at quarries, for example, wastewater treatment plants, airports, industrial plants, and also the inner city of Melbourne, which is indicated here with this square. This square is magnified here, and there you can see that we were going for different rivers and lakes as well. For time reasons, I'm just going to talk about one river, which is the Yarra River, and that's this one indicated with these purple needles. So when you do environmental analysis, you are basically facing two main challenges. The first challenge is related to the fact that you generate a vast amount of data. So in this case, for example, we were looking at 70 different locations. We did triplicate analysis, looking for different elements. We did screenings, we did calibrations, blank analysis, and at the end of the day, we ended up having 4.5 billion data points. So obviously that's something that you cannot handle yourself, and we need help by software. And in our case, we programmed our own Python-based software. In this case, we developed a graphical user interface which incorporated all kinds of state-of-the-art thresholding algorithms, calibration algorithms, and some additional features as well, which allowed us to identify particles, to mask everything that is not important, and then to do the calibration to, for example, investigate sizes, masses, and particle number concentrations. All of that is automized. We can do batch analysis, we can exclude regions of interest, and we can do much more than that. And here you can see a small video how it actually works. You can see that by just entering some experimental parameters, we can easily calibrate the sizes like this. We can also load, for example, reference files like reference particles to do calibrations, and it actually turned out to be quite straightforward with this program. So with this program, we actually could analyze the whole data set with a quite good computer in a bit longer than 5 minutes. That was the first challenge. The second challenge is related to actually identifying what kind of elements are associated with particles. And that sounds easier as it actually is, because the issue here is that you have to give some time for each element to see if we have, oh, sorry, that was too fast. We have to give some time to each element to see if we have this typical single particle data pattern. And in many cases, you just see some noise because there are no particles. And we actually have to screen through the entire mass range for each sample to see if we can find any kind of particle in there. So it takes a long time to actually find something like, for example, here now we can see that we have some typical single particle pattern for gold. So we actually used our bandpass mode, and the idea was here that we can define a larger mass window comprising several masses, and this mass window was then moved quite quickly over the entire mass range, and that allowed us to pick up signatures a bit faster. As soon as we had like a hit, we went back to the range where we detected this pattern, we established unit mass resolution and did the calibration. And this method alone actually sped up everything by factor 4. Okay, so once you did the screening, you can actually identify different kinds of nanomaterials. And today I'm just going to talk about one, which is lead. So we found that lead is associated with particles. Again, here we're looking at the Yarra River. This is the largest river in Melbourne, and from my point of view, it's also the most interesting one because it is actually associated with a quite interesting history of European settling. So the industry has been built up next to this river. There's lots of agriculture in the catchment of the river, also due to the gold rush a couple of decades left ago. There's some mining in the proximity. There's a waste management that doesn't always work. We have stormwater coming in, and we also have the influence of littering, for example. So overall, this is quite an interesting river from the point of view from an analytical chemist. If you want to swim there, it's maybe not the best river to pick. So here you can see the geographic course of the river. And in our case, we wanted to determine our, our lead-based particles. So we started outside of Melbourne and then worked our way downstream toward the so-called Port Phillip. So in this case, you can see, for example, here some raw data. We can indeed detect this lead signature for particles. And here you can see the particle number concentration. You can see that until sample location number 7, we have quite high concentrations, and suddenly we have a decline. We also determined the ionic lead background as well as the mean mass of lead per particle. So in this case, you can see that something is happening around here. And the question was, what's going on? We actually had no idea. And we found out by looking at Google Maps. And there we found that there's a quite tiny waterfall, which is separating the entire stream into an upstream section and a downstream section. Here we have fresh water, and down here we have the influence of the tidal seasons. So salt water is coming in twice a day. It's changing the matrix. We have basically brackish water, and this has consequences for the stability of the particles. And we can actually just look at the raw data in this case. You can see here, for example, sample location 7, sample location number 11, and you can see that as soon as we are in salt water, we don't detect anything any longer. We can also simulate that. We can, for example, take a sample from location 7, dilute it once in pure water and once in seawater, and you can see that after following dilution in seawater, everything disappears, while in pure water, we can still detect these particles. If you look very closely, you can see that another thing changes upon dilution. Here we have quite high signals. As soon as we dilute it, the intensity actually declines. And this is indicating that the particles must lose some lead. So lead is being released and they actually become lighter. And that made us wonder a bit what's actually going on here. And we started to think that we probably don't have discrete lead-based particles, but potentially some natural particles and ionic lead that is holding onto it. And that's something that we wanted to investigate further. In this case, we used transmission electron microscopy paired with an energy dispersive X-ray module. So we basically looked for the needle in the haystack. We identified some nanostructures in our samples, and then we looked for the different elements which are associated here. And here we found group 1 and 2 elements. We found silicon and oxygen, but also a little bit of lead. We can also plot the data, and there you can see that we probably have here some silicon dioxide, and on the silicon dioxide we have this lead flakes. So we believe that we have ionic lead that is holding onto this silicon dioxide. Using our single particle results, we can actually also determine how much lead we have in average per particle. And here we're talking about roughly 3 attograms of lead per particle. We can also count the atoms, and here we're talking about 7,000 atoms in average per particle. So you can see single particle is not only interesting for describing engineered nanomaterials like upconversion nanoparticles. We can also look at incidentally produced or naturally produced particles in the environment and see how they actually behave under anthropogenic pressure, for example, in this case, when there is lead contamination. In the third and last part of my talk, I would actually stray a little bit away from the single particle analysis and show you that we can also use ICP-MS paired with laser ablation to also investigate different kinds of microstructures. In this case, we are investigating structures in biological tissues. So the procedure is actually quite straightforward. So we can take a sample of interest, for example, a brain. We can cut the brain, and then we mount the thin sections on a microscopic slide. Next, we take a laser ablation, hook it up with an ICP-MS system, and then we basically start up the laser system, place our sample in the ablation cell. We move the laser across the sample, and then bit by bit, we evaporate small amounts of our sample, and the resulting aerosol is transported into the plasma for elemental analysis. So we can just pick different elements that are of interest. In case of brain tissue, iron, copper, and zinc are always interesting. And then we can reconstruct an elemental image. So in this case, for example, you can see the hippocampus of a rat, which actually has quite defined microstructures. However, in many cases, we are not only interested in elements, we also need information on the proteins. And here it's actually quite interesting to study how elements are interacting with proteins. And that's actually a gray area at the moment, which is quite hard to tackle. But we can use ICP-MS as well, and we can use a small trick. And this trick involves antibodies. So we can take antibodies which are specific for a certain protein. We can label that antibodies with, for example, a nanomaterial, for example, ytterbium in this case. And then we can incubate our labeled antibody with our sample. The antibodies then selectively find their target, and we can afterwards use our laser ablation system to spot the labeled metal as a proxy for the antibody and its target. So in this specific case, we were interested in an enzyme called tyrosine hydroxylase. That's a quite important enzyme in the brain. It is actually responsible for the formation of dopamine. And that is in so far interesting because when dopamine and iron come together, they often form reactive oxygen species. And this is believed to be one mechanism in Parkinsonian disease. And we wanted to actually investigate what's happening. And in this case, we had a mouse model where we studied that. So here you can see again the distribution of iron, copper, and zinc. And if you now try to target the ytterbium as a proxy for the tyrosine hydroxylase, you can see the following. So you can see with a little bit of fantasy, in the hippocampus, we also have some ytterbium, which is indicating us there is some tyrosine hydroxylase. But the contrasts could be better, right? You might guess where I'm heading with this. We can use our bandpass mode to improve that again. However, we have a challenge here because you saw that as soon as you switch into the bandpass mode, we lose unit mass resolution. And we still want to analyze iron, copper, and zinc, so we can actually not afford to lose unit mass resolution in this range. And we can actually tailor a method to that specific case. So here you can see the stability diagram for a low mass element, mid mass element, and a high mass element. And typically the quadrupole uses this scan line to provide unit mass resolution over the entire mass range. So we can manipulate that in a controlled way. And in this way, we can actually get the typical bandpass effect in the high mass region, but we can do it in a way that we still contain the unit mass resolution in the mid mass region. And in the low mass region, we're actually getting higher mass resolution. Here you can see a typical mass spectrum. So you can see that in the lower mass range, we actually get quite sharp peaks. In the mid mass range, around iron, copper, and zinc, we have unit mass resolution. And in the high mass range, we actually get this typical bandpass effect that we wanted. So here you have the comparison. This was the standard quadrupole method. Switching into the bandpass mode, we can improve ion transmission and therefore contrast. We can now study the microstructures and see how the microstructures incorporate tyrosine hydroxylase. But more importantly, we can also study the different endogenous metals. So in this case, as I mentioned, we were looking at iron and tyrosine hydroxylase as a relevant pair in Parkinsonian diseases. And in this case, you can see in blue the ion distribution. In yellow, you have the tyrosine hydroxylase resolution. And here you have the correlative picture. And anything that is red here is indicating that we have a high correlation of both. You can see that we have here some correlations and here some correlations. And this error here is quite interesting in particular because that's the substantia nigra, and that's thought to be one of the origins for Parkinson's disease. So in this case, we had a mouse model and we could actually show that we can use the information on iron and tyrosine hydroxylase as a predictive index to estimate what kind of oxidative damage is happening in the brain and how likely it is to have neurodegeneration. So you've seen that we can use it for any kind of brain tissue, but of course we can study any kind of biological tissues. And this is the last part of my talk today. So we were also interested, for example, in other tissues. And one area that is quite interesting to study different microstructures is cancer, because we always have the tumor, and around the tumor we have this microenvironment, which is actually defining a lot of properties of the cancer. This was a collaboration with Spain. And in this case, we looked for one specific protein called MMP11 in breast cancer. So in this case, MMP11 is a protein which is responsible for basically degrading extracellular matrix. And that's important for the tumor to actually make space to grow. So it's kind of logical that if you have a lot of tumor growth, you often also expect to have some MMP11 around. So in this case, we actually wanted to see if we can look at MMP11 as a predictor for metastatization. And in this case, we had different cohorts. We looked at patients with metastatic breast cancer and with non-metastatic breast cancer, and we saw that whenever the cancer is metastasizing, we have lots more MMP11, which gives us an indication what the tumor is up to. In many cases, it's not enough to just say, this is high, this is low, and in many cases, especially when you do longitudinal studies where you have several days of analysis, it's quite hard sometimes to compare intensities from day to day. And that's why we wanted to move this into a more quantitative direction. And in this case, we developed a workflow to actually quantify the MMP11. And I'm not going to go into much detail here, but we basically developed a thresholding algorithm, which is basically picking up areas of interest. It is masking anything that is not related to tissues, and it can even spot the tumor if you use the right algorithm. So in this case, for example, we can see Otsu's method, which is actually a method from photography to distinguish contrasts. Next, we have to somehow translate the intensities from the ICP-MS instrument into in concentration, and here we can use external standards. In this case, we developed new workflows for gelatin. So we actually developed a new generation of mold-prepared standards, which are quite homogeneous and easy to use. And in the final step, we actually had to find out how many elements associated to one antibody. And in those cases, we used size exclusion chromatography coupled with ICP-MS that helped us to isolate the labeled antibody. In this case, we used the sulfur signal integral to the antibody and compared it to the labeled metal. And by doing that and knowledge of the primary structure, we could actually determine how many elements or isotopes we have per antibody. And in this case, we always had like around about 100. So that information actually allows us to move everything into a quantitative direction, and we are just about to publish this. It's currently under review. In this case, we did a quantitative study. We again had 3 different cohorts. We looked at metastatic breast cancer patients, non-metastatic breast cancer patients, and we also had control samples. And the pictures are quite similar as before, but the main difference is that we actually have here a scale that is indicating concentrations. So based on that concentrations, we could make some comparisons. We had, I think, overall almost 30 samples. And then we compared the different levels and could show that for metastatic breast cancer, we actually have significantly higher MMP11s than for non-metastatic and healthy control tissues. So with that, I'm coming already to a summary and to conclusions. I hope I could show you that ICP-MS has lots of interesting facets, which are interesting for example, characterizing different kinds of nanostructures, microstructures, and also, for example, anatomic biological structures, which are relevant in pathologies and health. We can use dedicated methods, like, for example, the bandpass mode to accommodate specific situations, for example, to analyze labels as well as endogenous elements. We can do imaging. And finally, we can apply all of that for biomedical analysis. And with that, I'm coming to my acknowledgement slide. Again, thanks to the DIS Fachgruppe and for the award. Thanks to my PhD students, the different institutions which supported me over the last years, funding organizations, and lastly, also thanks to you for your kind attention. I think there's no time for questions this time, I don't know. And so you can ask me, if you have questions, you can approach me in the next break, and I would be happy to answer some questions. Thanks.
Congratulations again, and well-deserved award. And as you could see, many brilliant results. So well-deserved, and thanks to the Fachgruppe for doing this.